this goes into the functional aspects of the cardiovascular system and really getting into the electrical activity of the heart here. Okay, so objective 2 is to understand the electrical properties of the heart, really how energy and impulses are transmitted through the heart. And the first thing we'll do is describe the nature of the cells, some of the cells of the heart. They have this autorhythmicity, and the cells of the heart are able to generate their own rhythm. This is basically allowing for the pacemaker ability of these cells. They can provide a pathway for the spread of excitation, so the spread or conduction of this electrical activity, because this electrical activity is being spontaneously generated from these two groups or these two clusters of cells. So that is the pacemaker cells and then the conduction fibers, okay? So these cells make up a smaller percentage of the heart, and they are responsible for setting the rhythm, setting the rate of the heartbeat as we know it, and really determining the initiation of that electrical activity. We then have other groups of cells, which we will talk about going forward, but these make up the majority of the cells. These are actually contraction cells, excuse me. So the contractile cells are responsible for contracting. That is allowing for the force generation of the cells, of the ventricles and the atria that actually contract. But in terms of the electrical impulse, that is propagated by the autorhythmic cells, which are these cells here, the pacemaker cells and conduction cells. So let's talk about these groups of cells and look at their differences. So the pacemaker cells can spontaneously depolarize. They can just, through fluctuation of the membrane potential, they can spontaneously generate an action potential. And we would have talked about this property before. We also see this in single unit smooth muscle. So it has this inherent property to just kind of manipulate the membrane potential and spontaneously, without any neurotransmitter, without any neurons or neuronal input, they can spontaneously on their own generate an action potential. Because of this autorhythmicity, they can coordinate the rhythm of the heartbeat. So when the pacemaker cells fire an action potential, if they're firing at 70 beats per minute, that is going to be the heart rhythm, right? It's going to be 70 beats per minute because that is the rate at which electrical activity is being initiated, all right? The conduction fibers are similar. They have the ability to rapidly conduct action potentials by the pacemaker cells throughout the entire myocardium. They do have autorhythmicity of their own. So for example, if our pacemaker cells were to give out, the conduction fibers could help out with the rhythm, but they initiate action potentials at a much slower rate. So they're not as good in terms of that rate and that rhythm as the pacemaker cells, but they do have some spontaneous action potential capability. And their main role is really for propagating that action potential that is initiated from the pacemaker cells. Let's look at the examples or the actual groups of pacemaker cells. So we've got the sinoatrial node, known as the SA node. This is the main pacemaker of the heart. This is going to initiate action potentials at the fastest rate. This is essentially the captain in chief, right? This is the one that's setting the tone, setting the weight, because it can initiate action potentials at the fastest rate that we'll see. The second group of pacemaker cells are the atrioventricular nodal cells, or the AV node. These can also generate action potentials, but they're going to, as I said, do it at a slower rate. And so if the SA node is having trouble, or if there is loss of blood supply to the SA node, the AV node can certainly take over, but it's going to provide a much slower heart rate and rhythm than we see if the SA node is initiating those action potentials. And then the conduction fibers, we've got 3 sets or groups of fibers that help to propagate that electrical activity. They are the internodal pathways. The internodal pathways communicate from one node to the other, hence that term internodal. So they're going to send signals from the SA node down to the AV node, really sending that electrical activity from one group to the other. The bundle of His is going to be propagating from the AV node down through the ventricular septum, which is the central part of the ventricular wall. And then from there, the signal is going to go to the Purkinje fibers, which are along the inferior aspects of both ventricles. Let's look at the actual firing weight of these groups of cells and these conduction fibers. So the SA nodal cells have a firing weight at 70 to 80 action potentials per minute. And this just means that this has the fastest rate of firing. So if 1 minute has elapsed, the SA node is going to fire 70 to 80 action potentials. And in that way, it's going to be able to set the rate for what the heart, set the rhythm for what the heart rhythm will look like. The AV node is going to fire at about 40 to 60 action potentials per minute, the bundle of hairs at about 20 to 40 action potentials per minute, and the Purkinje fibers also at about 20 to 40 action potentials per minute. So as we can see, as we go down the chain here, the rate, the firing rate gets much slower. And so there are cells that can take over this ability to start the rhythm, but the SA node is the best at this because it has the fastest rate of doing so. Now, cardiac myocytes and cardiac cells are generally linked through gap junctions. So another, another note of detail here for your Venn diagram as you're thinking about which cells, which muscle types that is, have gap junctions. So single unit, which we spoke about already, and then gap junctions are also seen here in cardiac muscle as well. They have the fastest depolarizing cells that help to control the other cells. So I talked about the pacemaker cells, and then we also have the contractile cells. So the rate at which the SA nodal cells are firing is what controls the speed with which that electrical activity, that electrical impulse can be propagated down to the other cells and then can reach the contractile cells and result in muscle contraction. And so that's why we call these the pacemaker cells. These set the rate, set the tone, set the heart rate that we know it as about 70 to 80 beats per minute. Now, what we'll go on to see further down when we look at the cardiac output and how that is regulated with the autonomic nervous system is that if I were to take an SA nodal cell and put it into a petri dish, so put it outside of the body in vitro, in a petri dish, I would see that it would actually fire action potentials at about 100 beats per minute, 100 action potentials per minute. And so because of the influence of the parasympathetic system, so we made this point when we spoke about the autonomic regulation that at rest, the parasympathetic system is slightly more dominant than the sympathetic system. So because of the influence of the parasympathetic system, somewhat slowing down the SA nodal firing rate, what we see is that the SA nodal inside the body fires at about 70 to 80 beats per minute, which is how we get our resting heart rate, right? 70 to 80. But if we were to take it out of that parasympathetic influence or outside of the body, it would actually be much faster at about 100 beats per minute. Okay, let's look at how the excitation or impulse is spread among cells. So we start from atria. The atria fully contract, and then the ventricles fully contract. So kind of going back to that point that I made before when we were looking at those practice questions, that the coordination of the activity that's happening throughout the heart, throughout the cardiac cycle, really relies on the atria doing one thing first, right? They're spilling that blood. As they contract, they spill that blood from the atria down into the ventricle, and then we have to have the AV valves close. Then the ventricles can contract and eject that blood. So there really is a cyclical coordination such that the atria do their thing, they eject the blood down into the ventricle, AV valves close, then we have the ventricle doing its thing, the semilunar valves are now open, and the semilunar valves will close. So there's that atrial contraction, ventricular contraction in a very coordinated fashion. This is due to the presence of gap junctions and the conduction pathways, the conduction fibers, that is the bundle of hairs, the Purkinje fibers, the internodal fibers, really helping to spread that electrical activity from the SA node where it's starting all the way out to our other fibers and eventually to the contractile cells. And the gap junctions are what help to coordinate that, that propagation, that spreading of the activity. Another important feature that we see as a part of cardiac muscle are the intercalated discs. And the intercalated discs are a junction in between adjacent myocardial cells. So this area where two myocardial cells are coming up, or the cells are stacked against each other, so we have that small space in between, there is a junction called the intercalated disc. At this junction, we have the presence of those gap junctions that allow for the electrical activity to pass from one cell to the other. There was also the structure called a desmosome, and a desmosome is a type of tight junction that really helps to reinforce the cardiac cells as they are stacked against each other and really prevent any mechanical tearing or shearing, any stress that can be put on these myocytes as the heart continually contracts and relaxes, okay? So the heart is one of those very few tissues that is always working. It's not like your biceps or triceps that have an opportunity to rest and relax. The cardiac myocytes are always going continually, right? It's synonymous with life, with the circulation of oxygen and nutrients through our circulatory system. So it's never taking a rest. And because of that, it really needs the reinforcement with these desmosomes to prevent any overexertion, any ripping or tearing that would possibly happen because of the continuous work that these cells have undergone. Okay, just illustrating some of that's happening here. So we've got the SA nodal cell, which is our pacemaker cell. This is the one that's setting the tone, setting the rate, setting the rhythm. It will then send impulses to the other cells via the intercalated discs. So what we can see here is that we're sending the impulse from one cell to the adjacent cells via the opening in between, that is the gap junction, which is present at the intercalated disc. If we were to look even closer at what's happening at the intercalated disc, we would see the two structures that we just mentioned. So the gap junction, allowing that impulse to go from one cell right through this structure to the other. So a means of communication, very similar to what we see in terms of that single unit smooth muscle, allowing cells to speak to each other. The second structure that's found at the intercalated disc is the desmosome. And we can see how the desmosome is anchored to one side of this myocardial cell. And then it's anchored onto the other side at the adjacent myocardial cell. And so it's kind of strapping these two cells in really tightly together and preventing them from coming apart, from ripping or tearing or any type of injury, and really reinforcing that cell-to-cell junction. Now, what I want to point out here before we move along is that the other aspects of the muscle are pretty much the same. So apart from these structures at the intercalated discs, we have the same structures that we saw on skeletal muscle. So we have the myofibril that's forming these sarcomeres, so these elongated structures that give skeletal muscle and cardiac muscle its striated appearance. And then we have the actual axon and myosin filaments that are being anchored at each sarcomere. We still have the Z discs. We still have the regulatory proteins, so tropomyosin and troponin, playing a regulatory role along with calcium. So all of that is pretty much repetitive as far as the structure of skeletal muscle and what's happening here with cardiac muscle. Okay, let's take a pause here and see what questions we've got so far, and then we'll get into the order of conduction as far as that electrical activity. Okay, now what we'll look at here is what happens from the time the pacemaker cells initiate that impulse all the way until that impulse goes all throughout the heart and comes all the way up to the valve. So this is called the conduction system. So the action potential is initiated at the SA node. That signal is then going to spread via the internodal pathways. It's going to spread to both atria first. It's going to spread across. So the SA node is actually located in the right atrium. And I'll kind of point this out on a future slide, but it starts in the right atrium. And then that impulse travels across the interatrial septum and engulfs the entire left atrium as well. So that allows for both atria to fully contract before the impulse starts going lower toward where the ventricles are. After both atria contract, when that impulse is engulfed throughout both of the atria, then we travel down through the internodal pathways and toward the AV node. So the AV node is actually found within the ventricular septum. So that wall that separates the right ventricle from the left ventricle, this is where we have the AV node. So we go from the atria down through the internodal pathways or the internodal fibers, and then to the AV node. From there, we then go from the AV node down through the bundle of His, also called the atrioventricular bundle, which goes right down the middle of the septum. And then the impulse splits off. So the impulse splits into left signals and right signals along the left bundle branches and right bundle branches. From there, it's then going to go to the Purkinje fibers, and the Purkinje fibers go up towards the top of the ventricles. This is where the valves are located. Okay. All right, so just an illustration here to sort of make that make a little bit more sense. Let's walk through the 5 steps here of how that activity is propagated. So we start with the impulse at the SA node. And the SA node is located at the top of the right atrium, so sort of in this general direction here. From there, it spreads to the right atrium and very quickly across the septum of the atria, all the way over to the left atrium. Okay, it's really vital for the atria to receive the impulse first so that they can contract, squeeze that blood down, and then after that blood has filled into the ventricle, we can have the atria relax and the ventricles can start their contraction. This is why the impulse must jump across to the left atrium, allowing for both atria to fully contract. It then goes down to the AV node. So the AV node is somewhere about here. So kind of in the septum of the ventricles here, where the wall separates left and right ventricles. And it has to travel through the internodal fibers. So the internodal fibers are in between the SA node and the AV node. And this is how the impulse gets down toward the AV node. From there, the impulse is transmitted to the bundle of His. And the bundle of His is actually within the ventricular septum. So the wall of the ventricle here, we've got these fibers that are running from top down to bottom. These are called the bundle of His fibers, also known as the atrioventricular fibers or the atrioventricular bundle. Okay, from there, we split off into right and left bundle branches. So the left ventricle has left bundle branches, the right ventricle has right bundle branches, and this is how the impulse splits off as it gets to the bottom of the ventricle and then starts working its way up toward the top of that ventricle. Then the Purkinje fibers take over. So from the right and left bundle branches at the very bottom of the ventricle, the impulse is transmitted to the Purkinje fibers, which takes that impulse right up to where the valves are located in between the atria and ventricle. And the reason that and so at this point, the ventricles will contract. So after both atria contracts, the impulse comes down the center of the heart, then both ventricles can fully contract. The reason that this happens this way, the reason that the impulse goes down the center first and then engulfs the ventricles going in an upward fashion is to allow for that blood to be ejected. So in a similar way that you would squeeze a tube of toothpaste, you would squeeze it from the bottom. This is why the impulse has to go all the way down to the bottom first, so that it starts from the very tip, the very ends of the ventricle, it starts contracting its way up, and that allows the blood to be ejected from the chamber rather than the entire chamber squeezing and kind of trapping that blood inside. So in the same way that you would roll your tube of toothpaste up from the very top, from the very bottom up to the top to really allow for those contents to be expelled from the top, is the same way that the impulse engulfs the bottom of the ventricles first and then works its way up to where the valves are, squeezing that blood up and out into the artery. Okay. All right. Now this little walk through here, this little representation here shows how the impulse moves. So I'll use this to kind of go over that one more time here. So we start with the SA node, which is right here on the top of the right atrium. We then spread the impulse right the way across, all the way over to the left atrium as well, by the interatrial pathway. That allows for both atria to fully squeeze and contract, squeezing that blood down. The AV valves are open at this point, semilunar valves are closed at this point, and blood can be ejected from the atria down into the ventricles. It goes to the AV node, the impulse the impulse goes to the AV node and then out through the atrioventricular bundle, also called the bundle of His. So these fibers that are moving down the center here. The impulse now moves to the right and left bundle branches. So the fibers split off here. We have some going to the right side, some going to the left side here, right side here. And then we get into the Purkinje fibers. So the Purkinje fibers are going from the base of the ventricle all the way up toward the valve. We can see that these fibers end right here at the valve, which is where we're trying to send that impulse, all right? And so we see that the atria fully contract, the impulse goes down the center of the septum here, and then the ventricles can fully contract from the bottom upwards.